So my name is Jay Dhonoa. I'm a third-year undergraduate at the University of Chicago. I work in the Department of Medicine, and my research centers around using machine learning to predict the likelihood of the flu outbreak. So when talking about, flu outbreaks, we have two main types of events. We have the seasonal epidemics, which are the year-to-year flus that you get vaccinated for, and then we have pandemic events like the 2009 swine flu outbreak. And the difference between the two comes down to regular versus novel viruses. So regular viruses are essentially circulating in a closed population. It's the same variant of the flu that you get every year, just with different mutations. But a novel virus is a virus that's completely new. It comes in from some external source, and as a result, is usually able to infect a population very well. Usually, by this we mean that it comes from animals like birds or pigs. So there are two elements of a virus that we're concerned about. The first is the ability of a virus to infect a host. So for example, many versions of bird flu and swine flu cannot infect humans because they target proteins that are specifically found in birds and pigs respectively. And the second is the ability of a host immune system to recognize the virus. So when you get sick, your body makes antibodies to bind to these little proteins on the surfaces of bacteria and viruses called antigens. And they're very specific to that particular antigen. But over time, bacteria and viruses randomly mutate. And some of these mutations will change the shape of antigens on the surface, which means that your body will no longer recognize them. And that's this thing called antigenic drift. This is the reason why you need a new flu vaccine every year, is because the outside of the virus has changed enough that your body no longer recognizes it from, oh dear. But your body no longer recognizes it from the previous year. So to sort of illustrate why vaccination is important in this context, right? So the idea is essentially you can teach your immune system to fight a virus, right? And that effectively gives you the immunity that you would get from getting sick and then getting over it. So the more members of a population are immune to a virus, the fewer people can get sick, and also the more slowly the virus spreads. However, in the case of a novel virus like the 2009 H1N1 virus, because it came from a completely external source, it's able to spread through the population because we just didn't know about it. And so the way this actually happens is this process called reassortment, where two viruses infecting the same host can sort of mix and match parts. So in the case of the 2009 virus, we had a virus that was able to infect humans, but some of its antigens were actually from pigs, which means that we hadn't been vaccinated for it and we hadn't been ready for it. And so then my work is predicting whether or not a virus can jump from one species to another. And so some sort of criteria for this are essentially, we need to be in contact with the species, obviously, and we also need similarity between the viruses because they need to infect the same hosts. So then what information do we have about the problem? We have, so every year, when people get sick, doctors will sample that virus and create a protein sequence of the virus. These viruses usually come with a date and location, so we can uniquely analyze sequences from China, for example, versus from the United States. And then we also have the actual recorded history of outbreaks. And so the approach is, we use machine learning to create classifiers. A classifier basically just tells you whether or not an object is of type A or type B, right? And we create classifiers that tell us whether or not a sequence belongs to a human or it can infect swine. We use HPC resources to generate, thousands of these classifiers across different years and locations. We identify parts of the virus that are actually important for its ability to infect humans, and then we use those to predict the likelihood of a jump. So this is an example of a classifier. It's called a decision tree. And the way it works is that, we'll look at the 191st protein. If it's one of these proteins, we'll look at this. But if it's one of these, then we'll just guess that it's a human virus, right? And once we look at this location, if it's one of these, we go down the left, and so on and so forth. And basically, using these, we can come up with a few different ways to measure the risk of a jump. The first is measuring, the complexity of the model. intuitively can think of this as the more similar two viruses are, the more locations we have to look at to try to accurately tell them apart. And as you can kind of see from the graph, we have a large increase in this in 2008 and 2009, which is when the actual jump event occurred. And the second sort of metric of jump risk is classification accuracy. So, we can build as many decision trees as we want, but even if the best ones aren't predicting well, that means that there is enough similarity between the two species that the likelihood of a jump is very non-trivial increases. So, we start out with around 95%, but over the years, we gradually kind of drop until 2006, 2007, 2008, where we sharply drop down to about 80% accuracy, which means that, for a fifth of the samples that our classifier is looking at, it just can't tell whether or not it's human or swine. And that's usually a good indication that, a jump event is likely. So using the date and time stamps, or the time and location stamps, sorry, we can, sort of create a heat map of the jump risk, essentially by location. And doing this, we kind of get back the results you'd expect, which is that as we approach 2008, 2009, we'd expect to see a sharp jump risk in China, because that's where the virus first emerged. And that's indeed what we get here. Due to international travel, we'd expect that some viruses from the human side circulate around the globe, and that's possibly a reason why we also see a corresponding jump in the risk in the United States and also Europe. So this offers us a potential major shift in. Sorry? Yes. Yes. Why did it originate in China?
Why is the risk higher in Europe? It originated in
I see.
Why is the risk higher in Europe?
Basically, this is sort of like a geospatial function, right? So the closer you are, so pigs don't travel all that much, right? But humans do. But essentially, so the pig populations will be relatively isolated, and the closer you get to, a particular population of the risk, the more likely you are to have populations of the risk. Also, another element of it is that the reassortment that initially led to the 2009 virus was from Eurasian strain of the flu. So we would also kind of expect Europe to see a similar risk in the jump in risk. So, this leads to kind of a potential shift in how we predict the likelihood of outbreaks, right? Because essentially what this means is we can predict the 2009 outbreak, a few years in advance using this technique. And one of the remarkable things about this is that it's sort of domain knowledge agnostic. So when we're training our classifiers, right, they don't know anything about biology. They don't know how proteins fold or what sites are important beforehand or, how binding works between proteins. Right? They just get a series of letters. And, we just use statistics. And so one of the interesting parts about this is that we were able to rediscover locations that biologists had manually identified to be important on the virus. So this is the hemagglutinin surface protein, and the areas highlighted in green are sites that biologists have noted in research are important for its ability to infect different hosts. But our model also suggested new features that are highlighted there in red, which were features that, as far as we'd read in the literature, hadn't been noted as important for the virus to distinguish between, binding to a human versus binding to swine. And so, what are the sort of implications of this? The first and most major one is that, we have a method of potentially anticipating when a pandemic event like this happens. But the second is that, it's location specific. And so then by targeting areas that we see a major jump risk in, be it through vaccination or through culling of local animal populations, we can potentially take steps to mitigate the risk of these pandemic events happening in the first place. Just some acknowledgements. I'd like to acknowledge my lab mentor, for Dr. Shanu Chattopadhyay, for helping guide me through this work. I'd like to acknowledge the University of Chicago's Research Computing Center for giving me the computational might to tackle this problem. And I'd like to thank the folks at Lenovo for bringing me out here to talk to you guys today.
Ladies and gentlemen, Jay! I saw this.